everyone and welcome to Instant Biology by Dr. Neela. I'm here with you with another video, and in this video we would be talking about plasmids, and to be more specific we would be discussing about the types of plasmids, the functions of plasmids, applications, examples, and I will also be discussing one previous year question regarding plasmids, right? So I've already made a video on plasmids previously, and that video was in Hindi. So if you are more comfortable in Hindi, you can watch that video. Otherwise, if you are comfortable in English, then you can prefer watching this video. So plasmid is very important, and we would be talking about the different types of plasmids, the different PUC plasmids, the EB plasmids, and different types of plasmids we would be talking in detail in the upcoming videos, but today I would be giving you a general introduction about the plasmids. So let us not waste any time and let's get going. So coming to plasmids. Now, what exactly are plasmids? So the first question that is present on the screen also asks you the same thing: what are plasmids? So plasmids are nothing but they are most often circular double-stranded DNA found inside the cells. So they are circular. The first thing that comes to mind over here is that they are circular in nature, something like this, right? So they are circular double-stranded DNA. This is what I've drawn. So it is a double-stranded DNA, double-stranded and circular, okay, found inside the cells. Which cells? Basically they are present normally or generally they are present inside the bacterial cells. Bacterial cells. Now you will say, can you show us an example of that, or can you show us a diagrammatic representation of this? So you can see that this is a bacterial cell, and in the bacterial cell this is a plasmid that has been shown over here. So these are circular double-stranded DNA structures that have been shown over here. Now you might be wondering that what is this particular structure that has been drawn in red color? This is actually the bacterial DNA. When I say bacterial DNA, I actually want you to understand that this is the chromosomal material. Chromosomal material. Now, you might be wondering that is the chromosomal material or chromosomal DNA of a bacteria different from that of a plasmid? Because both are DNA in nature. So is this different from this, or are both of these things same? No. Sorry, understand this clearly that the chromosomal DNA is always different from the plasmid DNA. Plasmid is also called as extra chromosomal genetic material, also called as extra chromosomal genetic material. So what does extra chromosomal means? Extra chromosomal means that it is present outside the chromosomal material. It is not a part of the chromosome. So it is present outside the chromosomal material. This is what is called as a plasmid. So this is the first thing that we have understood, that they are circular in nature, they are double-stranded DNAs, they are present outside the chromosomal DNA, and they are majorly present inside the bacterial cells. Apart from that, we can see that they are not attached or associated with the chromosomal DNA. So this is what we have already discussed, that they have nothing to do with the chromosomal DNA, they are separate entities. Now, they are also called as true replicon. Now when I use this term replicon, what do you understand from this? Something that has the power or something that has the capacity to replicate, something that can form its own copies. So yes, plasmid has the capacity to replicate its own self, because there is a particular region in the plasmid called as origin of replication. Now let me draw it over here. So suppose that this is a region in the plasmid that is called as ORI region. ORI region. So what does ORI stand for? ORI means origin of replication. So since this origin of replication region is present inside the plasmid, therefore it has the capacity, or it can replicate or divide. Now this is what is meant by true replicon. Okay. So now we have understood about the first slide, I believe. Let us move on to the next slide. So the next slide says that it can be stably inherited in an extra chromosomal state. What does this mean? Stably inherited means that this particular plasmid can be taken or can be taken up by another bacteria understand this very carefully, that it can be stably inherited, can be inherited by another bacteria in the extra chromosomal state only. So suppose this is one bacteria, this is another bacteria. So this plasmid can be easily transferred from one bacteria to another bacteria, and this can be done very, very stably. It's not like that the DNA will be transferred from here till here and this bacteria will degrade that DNA. No, when the plasmid is transferred from here to here, this plasmid will be stable. This is what is meant in this statement, that stably inherited in an extra chromosomal state. Again, a very important point is extra chromosomal. That means it is not bounded with the chromosome. Next, naturally occurring in the bacteria. So as I've already said, that plasmids are generally seen inside the bacteria. Okay, so they are generally present inside the bacteria. Now, at times they are also present in eukaryotic cells. This is rare. There are certain examples wherein you can see that the plasmids are present in eukaryotic cells, but in general you will always refer to the prokaryotic cells or the bacteria whenever plasmids are being talked about. Next we have to talk about the functions of bacterial plasmid, but I would prefer talking about the types of plasmids first, and then I would be coming to the functions of plasmids. So let us now move on to the different types of plasmids that are over here. So starting with, what are the different types of plasmids? So obviously there are number of different types of plasmids, and we would be talking briefly about each one of them. So the first thing that comes to mind is F plasmid. What does F stand for? F stands for fertility over here. Let me just quickly write. F is fertility. What do you mean by fertility plasmid? Understand this, that you might be knowing that a good method of gene transfer from one bacteria to another bacteria is through the conjugation process, right? So there are different methods through which you can transfer the gene from one bacteria to another bacteria. Conjugation is one such method. Apart from that, there is also a method called as transduction, in which viruses are used to transfer the genetic material from one bacteria to another. And there is also a method called as transformation, in which naked DNA is used for transforming a particular bacteria or changing or modifying the genetic material of a particular bacteria. But here I am talking about conjugation. In conjugation, if I speak briefly, then in conjugation, the one bacteria, let us say this is one bacteria, it is connected with another bacteria with the help of a conjugation tube. This is called as conjugation tube. And through the conjugation tube, the plasmid of one bacteria moves into the other bacteria. Right? Through the conjugation tube, the plasmid of one bacteria will move into the other bacteria. Now, an important point to note over here is that the bacteria that will be serving as a donor of the plasmid is called as F+, and the bacteria that will be serving as a recipient will be called as F-. Understand this, that only when the F plasmid is present, then only the conjugation can happen, because the F positive or the F plasmid will help in making this conjugation tube and transferring the genetic material from one to another. This is the importance of F plasmid, so it will be responsible for conjugation. Okay, so there is a good example that is called as TRA gene or transfer gene. This is a very nice example. This is a very nice example of a gene that is present inside the F plasmid, and if this gene is present, TRA gene is present, then only the transfer will exist. And this TRA gene is present inside the F plasmid. So hopefully you would have understood that why F plasmid is useful in the process of gene transfer and why it is called as the fertility plasmid. Let us now talk about the R plasmid. So what does R stand for? R stands for resistance. R stands for resistance. What does resistance mean over here? Now, if R plasmid is present inside any bacteria, then what will happen? This R plasmid will confer resistance against any given antibiotic. Okay, now let us say that R plasmid is present which is having the property of resistance against ampicillin or against streptomycin, or let us say any other antibiotic. So if this R plasmid is present inside a bacteria, let me show it to you over here. Suppose this is a bacteria and this is having a R plasmid, right? This R plasmid is providing resistance against ampicillin antibiotic, ampicillin resistance. Now, if you keep this bacteria in the antibiotic ampicillin, what will happen? This bacteria would not die. This is what is written over here. So it carries genes for resistance to antibiotics. Okay, so I believe you would have understood. So there are different types of R plasmids. One example is RP4 of Pseudomonas. So inside Pseudomonas, this RP4 plasmid has been seen, which confers resistance against many antibiotics. This is clear about the R plasmid. Now coming to the col plasmid. What do you understand by col plasmids? Col stands for coli. Basically, this is produced or this is seen inside the E. coli bacteria, and it codes for colicin. Now what is this colicin? Colicin is basically a chemical. Colicin is basically a substance, or a toxic substance rather, we can say that. It kills sensitive E. coli and provides immunity to a particular colicin. Understand this. If a E. coli is having a colicin plasmid, or col plasmid, if a E. coli is having a col plasmid, then it will be able to secrete some toxic substances that will be killing the susceptible E. coli that are present in near vicinity. And also this col plasmid will also safeguard this particular bacteria against the colicin that is produced by other E. coli. Okay, so I believe you would have understood that suppose this is a bacteria that is having the col plasmid, it will be secreting some colicin, and therefore the other bacteria that are around, susceptible bacteria, they will be eliminated, killed. And if another bacteria is present which is secreting the colicin like this, there will be no effect on this particular bacteria because it is again having a col plasmid. Okay, I believe you would have understood this. Now coming to the degradative plasmid. This is another kind of a plasmid called as degradative plasmid. What is the purpose or what is the function of degradative plasmids? You might have seen that for the transfer of oil from one port to another port, large or big ships are used, right? So the ships, they transfer the petroleum or oil from one port to another port. At times what happens is that because of an accident, what happens is the ship gets overturned, and because of that large oil spills happen on the sea, on the sea waters. So because of that, a large loss of flora and fauna exists at that particular place. So how can we stop that? Because you see that the animals that are living inside the sea, they have only one source for the oxygen. So because of the atmospheric oxygen gets exchanged from the sea water, through the sea water, what happens is that the animals that are present inside the sea, they have oxygen to breathe. But if oil spillage takes place, oil spreads over the entire surface of water, now the gaseous exchange will not be able to take place, and this will result in killing of the organisms. So this is why something needs to be done for that. Now there is a very nice example of a degradative plasmid that can be used over here. So degradative plasmids can metabolize unusual molecules like toluene and salicylic acid. So these degradative plasmids will allow a bacteria or an organism or a prokaryote to degrade certain metabolites that are unusual, that will not be metabolized by any other organism. So they can metabolize these, toluene and salicylic acid can be metabolized. So this is a very nice example. The example is tall plasmid, and it is seen in Pseudomonas putida. Pseudomonas putida has this tall plasmid, and it confers or it allows this Pseudomonas putida to feed upon the toluene and salicylic acid and thus degrade both of these. So that is why it is also termed as a super bug in some cases, so that the oil spillage can be cleared. Okay, so I believe you would have understood this example also, or this type also, degradative plasmid. Now coming to the next type of plasmid, this is called as virulent plasmids. What are virulent plasmids? What does virulence actually means? Virulence means the capacity to cause a disease or capacity to cause pathogenicity, causing disease, right? So if a virulence plasmid is present inside the bacteria, then that particular bacteria will definitely cause some disease in the host organism, right? There are more chances that the virulence plasmid, if present, it will cause disease. Non-virulence, or if the virulence plasmid is not present, there are chances that no disease will be caused by the bacteria to the host. This is what is written. It confers pathogenicity on their host bacteria, right? So what are the examples? What are the examples of this type of virulence plasmid? So example is TI plasmid and RI plasmid. So TI plasmid, you might have heard about this, or you might have studied about this. TI plasmid is actually tumor inducing. Tumor inducing. It is seen inside the Agrobacterium tumefaciens, and this RI plasmid is found in the same species Agrobacterium only, and the same genus Agrobacterium only. The species is Rhizogens, and in case of TI plasmid, the species is tumefaciens. You should be knowing this. So TI and RI plasmids have the capacity to cause diseases inside the plants. You might have seen that Agrobacterium causes the nodulation, and RI plasmid will cause the crown gall disease. Okay, crown gall disease, and crown gall disease is caused by the TI plasmid. Let me write it over here. Crown gall disease, and RI plasmid causes hairy root disease. So both of these plasmids can cause diseases in plants. So I have written the names of the diseases. Now coming to another method of classification of the plasmids, the plasmids can also be classified on the basis of single copy plasmid and multi-copy plasmid. What does this mean? In simple terms, whenever a plasmid is or can make multiple copies, so this is called as a multi-copy plasmid. Now in case of a multi-copy plasmid, it has been seen that different control than the host cell, more than one replication per replication. This means that it is not dependent upon the bacterial replication. That means the plasmid is present inside the bacteria, agreed, but its replication will not be dependent upon the bacterial replication. That means bacteria can divide, let us say, one time, but the plasmid can obviously divide more than one times. This is also called as the relaxed replication method. Relaxed replication. But if the replication of the plasmid is only dependent upon the replication of the bacteria, this is called as the stringent replication. Obviously, if a plasmid is dividing, then only a bacteria is dividing, then it will be called as a single copy plasmid. That means only one copy will be present inside per bacteria. So single copy plasmid replication control is same as that of the host cell, because when the host cell is dividing, only then the plasmid is dividing. So you can say that replication control is same as that of host cell. It is also called as stringent replication. This is called as a relaxed replication. So I believe you would have understood this particular concept. Let us move on to the next slide and talk about the different functions of bacterial plasmid. So I believe most of the functions you would be able to understand now. The first is antibiotic resistance. I have already told you about the R plasmid. So they confer the resistance against antibiotics. Coming to colicin resistance, again you would have understood that we have talked about the col plasmids. They confer the secretion of the colicin, and whatever bacteria are present around this bacteria which is secreting colicin will be dead. Apart from that, there are certain bacteria that have the capacity to do the nitrogen fixation because of the presence of such kind of a plasmid. Next is symbiosis. There are certain bacteria that can do symbiosis because of the presence of a plasmid, specific plasmid. So again, symbiosis, nitrogen fixation, these are the characters that can be attributed to the presence of plasmids. Apart from that, tumor induction in plants. I've already told you that TI plasmid causes tumor induction and causes crown gall disease. Agrobacterium tumefaciens bacteria has this TI plasmid. which causes the disease. Next is fertility plasmid. I have already told you what fertility plasmid is. It is also called as F plasmid. Whenever F plasmid is present, it will cause the formation of conjugation tube and help in the transfer of gene from one bacteria to another bacteria. Right. One more important thing is that, I have told you that it is present inside the eukaryotic cells also, and in case of eukaryotic cell, plasmids can be of size 2 micrometer plasmid of yeast. Basically, it is seen inside the yeast cell when we talk about the eukaryotic cells. Now, since we have discussed this, I have also told you about these things. We have discussed this also and this also. Now, coming to some more properties of the plasmids. So, bacterial cells can survive without plasmid. This is a very, very important point and has been asked in different life science examinations, that it is not necessary that all the bacterial cells should be having plasmid. It is just that the cells that have plasmids have the added advantage over the other bacterial cells. It is not very essential for survival, but yes, it will give you some added advantages. This is the important thing. Plasmids provide growth advantage. This already I have explained to you. Cannot be considered as genome. I have already told you that the chromosomal material or the genetic material, chromosomal material is different from that of a plasmid. Why cannot we consider it as a genome? These are certain points that I have jotted down to explain it to you, that why it cannot be considered as a genome. Because it can mobilize. That means plasmids can be transferred from one bacteria to another bacteria, but the chromosomal material cannot be transferred. This is the reason why we can say that it is not a part of genome. Next, it can be present or it can be absent. That means plasmids are not essential for survival. They can be present, but chromosomes will definitely present. They are inevitable part for the survival of the bacteria. Next, not indispensable. This is what I have already told you. That means plasmids can be deleted also, but chromosomes, if you delete, then the bacteria will not be able to survive. Now coming to the size and shape of the plasmids. What are the different sizes and shapes? So the size basically vary enormously. Okay, from one bacteria to another bacteria, you will see a large variety of the sizes present inside the different bacteria. So the size of the bacteria basically ranges from 1 to 200 kilobase pair. This is the general variation of the size. Next, F plasmid. If I talk about the F plasmid of the bacteria, it is around 1% of the size of the E. coli chromosome. Right. So the entire chromosome, I have already told you that the size of the chromosome of E. coli, 1% of it is actually the F plasmid. If I talk about the multi-copy plasmids will be very small size, right? Multi-copy plasmids because there are multiple copies of the same plasmid that is present inside the bacteria. So if there will be multiple copies, then what will happen? There will be a constraint in the space. Obviously, these plasmids that are present in multi-copy form, they have to be in very, very small size. Then only they can be fit inside the bacteria. If you talk about the multi-copy plasmids, col E plasmid is a multi-copy plasmid, right? There will be multiple copies of a col E plasmid. So it is further smaller than the F plasmid because it is 10% size of the F plasmid size. Of the F plasmid only it is 10%, so it is further very, very small. That is why I said that there is a lot of variation in the plasmid size. In case of eukaryotes, a plasmid is seen that is 2 micrometer in size, right? Now, majority of the plasmids that you will observe would be circular double-stranded DNA molecules. I have already told you about this. Some would be linear double-stranded DNA molecules also. This is again a very important point. Not necessarily that always the circular double-stranded DNA can be seen in plasmids, but at some times you will see linear double-stranded DNA molecules, and this is seen inside some bacteria, this is seen inside some fungi, and this is also seen inside some higher plants. This is an important point, you should be remembering this. Not necessarily circular or double-stranded DNA. Now, RNA plasmids have been discovered. Not necessarily that always the DNA plasmids only will be present. This is again a very important point. There have been discovered, not only the DNA plasmids, there have been discovered RNA plasmids also that are seen in plants, fungi and animals. This is again a very important point that you should be knowing. Now let us move on to the next slide. On this particular portion or in this particular slide, a lot of PYQs have been, previous year questions have been asked. Let us just talk about this. So there are 5 different conformations. Let me just use my pen to make you understand this. There are 5 different conformations of DNA plasmids possible, which have different electrophoretic mobilities. What do you mean by electrophoretic mobilities? Electrophoretic mobilities means that if you are keeping that these 5 different conformations, then inside a electric field, then the mobility or the movement of these different conformations will be different. Okay, so you can differentiate between these plasmids on the basis of their electrophoretic mobility. Right, understand this, that there are 5 conformations, and I have arranged the 5 conformations from the top. That is, this one will be the slowest, and if you move down, this one will be the fastest. So what are the different conformations? Let us draw them. The first one is open circular DNA with one strand nicked. So this is the open circular DNA with one strand nicked. So this is a nicking that has happened, nicked. Only one strand has been nicked. Now let us move on to the next point. Relaxed covalently closed circular DNA. Relaxed covalently closed. Now this is the relaxed covalently closed circular DNA. Next, linear DNA. This is the linear DNA. Fully intact supercoiled DNA. This is the supercoiled DNA, basically. Fully intact supercoiled DNA. Next is supercoiled denatured DNA with unpaired region. See, this is the region that is unpaired, whereas this is a paired region. Understood this? So these are the 5 different conformations of a plasmid that can be seen, and they move and they have different speeds of their mobility if they are kept in electric field. Why is that? Because of the shape of these different plasmids. So if open circular DNA one strand nicked, it will be moving the slowest. Next, relaxed covalently closed circular DNA, it is closed, relaxed, will move a little faster. Then linear DNA will move a little further faster. Fully intact supercoiled DNA will move further faster, and finally supercoiled denatured DNA with unpaired region will be moving at the fastest speed. So this you should remember if you are giving any life science examination. It will be beneficial for you. So hopefully you would have understood the concept of the plasmids. I have a few announcements to make. Please understand this. So we have a new GATE biotechnology batch that is starting from the 23rd of July. So in that, basically in that batch, the classes will be taken in morning from 7 AM to 8:30 AM in the morning. There will be 1.5 hours class. In this batch, we will be providing a lot of things. You would be getting the test series, you would be getting daily DPPs, you would be getting recorded sessions, you would be getting notes and everything. This is a complete course. And we would also be doing 2 times revision at the end of the course. This is also a salient feature of the course. Now you would be getting the details of this course over here. That means at the top you can check out that, and in the description section also I will be giving you the details. You can contact with us on this particular number. So I am giving you the number over here. So it is 8299134528. 8299134528 is the number that you can connect with us on, and you can talk to us about this. Apart from that, we have another program that is for students who want to pursue their studies abroad, their masters or their PhD abroad. So for those students, we have a program called as UDAN program. In this program, we connect the students with the experts who have experience or exposure to applying abroad. Now we connect the students with these experts, and the cost is very, very affordable. This is a promotional cost since we have launched this UDAN program recently. So the cost is just ₹299 for a period of 45 minutes. You can ask the questions till 45 minutes to the experts and they will clear your doubts. Apart from that, we have services such as helping you in preparation of SOP or letter of motivation and things like that, that also you can use, but those are tailored programs and they have specific prices. Again, for connecting with us on Udan program, you can also again connect with us on 8299134528. You can WhatsApp us on this number and we will give you more information. I believe this video was useful for you. Do like the video and subscribe the channel, and do not forget to press the bell icon to get the latest updates. Thank you so much. Have a good day.